Good morning, everyone. My name is Cara Poon, and I'm a senior scientist at QuTEM. And it's my honor and my privilege to be talking to you all today about characterizing AAVs and other gene therapy vectors using transmission electron microscopy and other critical quality attributes as well. But first, I would like to give a short introduction to who we are at QuTEM. And some of you might actually recognize some of the materials that I'll be showing today, and that would be because we aren't exactly a new company. We have been around for quite many years, but under a different name, as Veranova Bioanalytics, and also as Veranova. In autumn last year, we were acquired, so now we have rebranded, and everything is now under the name of QuTEM. So at QuTEM, our main focus is to provide high-quality transmission electron microscopy services to the life sciences industry. And there are three core components to achieving this for us. The first of which is that we are a GMP-compliant lab. And if you have known us from before, you would know that Veranova Bioanalytics had a GMP certificate as well. So when we were acquired, we also reapplied for a new GMP certificate. And recently, we were also inspected by the Swedish Medical Products Agency. And so we will get our GMP certificate by May 12th at the latest. The second component to our services is that we have our own proprietary software. So it is an advanced software that we use as our database and also for image analysis. And it is also FDA compliant. The third part is that we also really heavily invest in our personnel. So I have the privilege of working with an amazing bunch of people who are not only experts in electron microscopy, but also in other fields such as virology and image analysis and quality assurance, for instance. And importantly, they also work with the same particles that you are dealing with on a daily basis. So we know all of the questions and problems that you are also asking about your samples. So if you have any questions about how to characterize your samples, please feel free to get in touch with us, because we will have someone who will be able to help you as well. So we are located in Stockholm in Sweden, but our clients are based all around the world, and they primarily work with drug and gene delivery and also with vaccines. They work with us together at all stages, from early R&D through to R&D, preclinical, clinical, and also the commercial phase. But what we really recommend is that they come to us as early as possible in their development so that we can, together with them, build a really robust analytical method that helps to monitor the critical quality attributes that they're interested in throughout the entire manufacturing process as well. So when they send their samples to us in Stockholm, what we do in our lab is then to prepare the samples, image them, and also perform the image analysis. And then within 10 business days, we will deliver the reports to them together with the images. And we can also do this faster than 10 days if needed. So then comes the question, why would you even want to consider using transmission electron microscopy as one of your analytical tools? Well, in my opinion, it's actually a really great method for understanding your design space. So if you have the example that you are working in the early phase, you haven't characterized your samples in detail, and you don't know exactly what you have, it's a really good way of actually seeing what's inside of your samples. So this means that also TEM can be a tool where you can see a lot of data. So importantly, it's then the responsibility of you and I to decide what quality attributes do you need to monitor throughout the entire process of your development. So these can be three questions like, what structures do you have in your sample? How much of them do you have? And can they be classified and also then correlated to important biological activities? So if we can answer these three questions for our clients, we are really proud that we can actually help them to build a really robust analytical method. There are also quite a number of different particle types that we look at QuTEM. And depending on which particle type you are working with, there are different types of quality parameters to consider as well. But irrespective of which particle type you have, the important thing to consider is that there are a core number of different quality attributes. And these are important to consider throughout the entire manufacturing process. So I want to talk about these in the context of AAVs today. And for instance, the first one that you might consider is then aggregation. Ideally, you want a sample that is not aggregated. You want all of your particles to be individual entities. And also, you want to ensure that your process has a good purification step so that you don't have too much debris, for instance, while also not purifying away your sample. You'd also like to then understand more about the formulation you have and also whether or not you have a stable process so that you are looking at the integrity of your particles over a period of time as well. Another attribute that you might want to consider is, say, have you incorporated the correct genome for your AVs? And this can indicate whether or not you had a suitable packaging process or not. So if you have a good understanding of all of these different attributes, for instance, you can use this information to make process-critical decisions so that you can make a really nice product which is both safe and effective, which is the ultimate goal. But, okay, images are nice, but we also need numbers. So for instance, you make a batch today, and you want to compare this batch to a sample that you made, say, 6 months ago. So we can then look at, for instance, aggregation of AAVs. And using our FDA-compliant software, what we can do is to detect out all of the individual particles, as well as clusters and aggregates as well. And then we can give this data to you in terms of numbers. What's the percentage of the individual particles that you have, and also what's the percentage of clusters and aggregates that you have in your AAV sample. Another very important critical quality attribute that you may want to look at is the AAV content. So by this I mean the percentage of empty, the percentage of partially filled, and also the percentage of filled particles. And what we do is that we look at the internal density of all of these particles in a data set. So in a data set where we have, say, 1,500 particles, we can plot them as a function of their internal densities. And in this representative sample, we can see that there is a peak for empty capsids, and what also looks to be a peak for, say, partially filled, and a peak for filled capsids. But the question also would then be, do you really have a nice sample where 86% is your intended genome? Or rather, did you have only 11% of your target genome? So this is a question that we have received from both regulatory authorities and also our clients. Can you actually determine what is in these different peaks in terms of their genome lengths? So this is something that we have now been looking into in more detail quite recently. And what we have found is that for every single genome length, there is a very distinct internal density signature, meaning that we can actually determine the genome length if we also know the internal density. So what we've done is that we have created a library with different samples of known genome lengths. We've measured the internal densities of all of them, and then plotted them, internal densities against genome length. And what we see is that there is a really nice linear correlation between both. So this is now our reference curve. And if we look again back at the data that I just showed you, now we can see what are the genome lengths of each of these different peaks. So on the left-hand side, we have an empty peak, which is around 0.2 kilobases. Then we also have the second peak, and we see this is 2.5 kilobases, and the third peak is 4.2 kilobases. So now this gives us a really good understanding of what's inside. And normally, you also know what your intended genome size is. So now, if we look a little bit more closely, this data now shows us that if our target genome was 2.5 kilobases, you've done a really great job. You have 86% of your sample as the intended filled peak. And you only have, say, 11% of the overfilled peak. This method has now also been going under a platform validation, and I'm happy to share some of our preliminary results with you today. So for the repeatability and the intermediate precision, we have quite a low standard deviation of less than or equal to 1% for determining the vector content, and by that I mean the percentage of empty, partially filled, and filled particles. And for genome length, we have a standard deviation of between 110 and 150 bases. What's really interesting, though, is the sensitivity. So by that, I mean how many particles do you need in order to be able to see a subpopulation? And this is only 50 particles. So this means that we only need to have 50 particles out of a data set of 1,500 particles in order to be able to tell you that you actually have a subpopulation present. And in addition, we also are looking at the resolution, and this is also quite incredible compared to other orthogonal methods, which is that in order to be able to differentiate between different subpopulations, the difference between them needs only to be 75 bases. So this is a really great technique if you want to be looking at several different critical quality attributes, and for AVs, for instance, you can look at aggregation, vector content, and also genome length. So QuTEM can also be used for other different structures as well that can be quite complex to actually characterize. And by that I mean we can look at lentiviral vectors, lipid nanoparticles, exosomes, and also VSVs. But what I really want to stress here is, again, you need to know which quality attributes you want to assess and monitor throughout the entire life cycle of your products. So I just want to give you two examples today, the first being lentiviral vectors. And perhaps you would like to know more about what particles you have based on their functionality, and by this I mean their morphology as well. So we use our FDA-compliant software to first detect out all of the particles, and then we can classify them based on the characteristic spike proteins that we see, which lend their functionality. And also, we also want to look out for if they have a high internal density, and also if they have the capsid inside. Other times, we might also see that the particles, say, have the characteristic spike proteins, but a very low internal density and no genome. And other times, we might see that they also don't have the characteristic spike proteins at all. So another case that we could look at is lipid nanoparticles. Here again, we use our software so that we can detect out all of the different particles, and we can then classify them out on their morphology. Typically, what we see is that they have either homogeneous low internal density or high internal density, indicating something about their genome inside. And also, we see that they are also compartmentalized. And these are all different features we can classify. And moreover, we can also measure their size and give you this data as a size distribution as well. So with that, I'm coming to the end of my presentation, and I would like to just leave you with a few takeaways. The first of which is that QuTEM can be a really comprehensive technique to give you a lot of information about different critical quality attributes. It can also be a really nice tool to add to your analytical toolbox. And importantly, if you know exactly which critical quality attributes are of interest very early on in your development, you can use this information to monitor your samples or your product development from the step of R&D, then choosing your formulation, all the way to release as well. And so this can be a really nice and robust technique. And if you have any questions, or if you have not seen the particle types you're working with, please feel free to get in contact with myself or my colleague Beata Hadavand. We also have a table out in the exhibition space as well. And thank you very much, and I wish you all a really nice day and a pleasant time at the conference.